Okay, so we'll have a brief panel session, so if the people who spoke could come up. Gene Kohler was going to do this. He's now arrived, but he's asked me to do this, and he'll do the one this afternoon. He's actually the co-PI on this effort to get funding. So, we have questions coming already. So go ahead. Just wait. Wait. And could you also say your name and where you're from? We've been asked to do that.
Jim Saluka, Indiana University. I like Dr. Karp's reference to Knuth and his literate programming. It's interesting, though, in the 30 years since he said that, it's changed. That he was concerned with the machine could understand the code, and there was lack of human documentation on it. And so he was going to embed documentation with the code. Now, there's a third reader. The machine has to implement the code. There should be human readable documentation, but there should also be a machine readable documentation of the biology that's in the model. So not only can a person look at that chunk of stuff and understand the biology being modeled, but another program can look at that chunk of stuff and say, I don't care what the computation is, I want to know what the biology being modeled is. So here we are 30 years later, we got a third player in the very elegant way Knuth put it, but now we've got to deal with this third player, that some other machine has to understand the biological concepts that are in that file.
I think that makes sense. I think that's another argument for the model residing within a database or drawing its information from a database. And, I argued for that databases enable multiple uses, and querying of the data associated with the model would be, for other purposes, would be another use.
So sort of just building on this theme, Peter, because I actually really personally thought that the notion of literate modeling is definitely a good one. I think something we might want to discuss in the open sessions, but it comes back to what was sort of alluded to a little in the previous panel, and that is, the problem, it seems, is the incentives. How do you incentivize people to do this? I mean, that's the key. I mean, I don't know if panel members or the audience wants to comment on that, because without that, it sounds good, but how do you get it to happen?
I think that is key. I think there's the subtle side, which is that if you submit your model for publication, reviewers are going to be more likely to peruse the model and more readily accept it if the model is published in a literate fashion and available in a literate fashion. I think the models will end up getting more widely used and disseminated if they're published and available in a literate fashion. But yeah, I'd like to hear ideas for more direct rewards that we could set up.
Do people have specific ideas about that?
I think it would be interesting to come up with a way to really reward reusage of a model you've put out, because currently, we've heard discussions already about the reproducibility of modeling results, but I want to make the provocative statement that most models are never being reused, and no one is interested in actually reusing them because they cover a particular cell state, a particular condition. They're being used for one paper, as was mentioned before. And nowadays we have a lot of possibilities to use standards like SBML or rule-based modeling to make our modeling results reproducible. But I think we have to work on incentives that bring people to the point where their modeling becomes biologically so interesting and valuable that people reuse the model because of the biological results the model was capable of providing.
So the experience with the genome scale metabolic models is exactly the opposite. They are easy to disseminate. Peter talked about even some web sources to set them up and compute. And I think because, for two reasons, they become widely used. One is that they are easily available and implementable. And number two, they actually have a strong knowledge base or database backing. And maybe number three, they actually generate useful results computationally.
Yep.
So those models have been widely disseminated and used, I think, for probably these three reasons.
So if we're trying to build a community here, we need to have as many voices speaking as possible. So
All
let's
right.
go to the... Yeah.
I have a question. Janet Palu from the College of Nanoscale Science and Engineering at the University at Albany. I wanted to comment on the view that biological information has to come from one system, because in fact, biologists or scientists of all sorts, we mix and match all the time, and that's actually where we make the greatest advancements. So certainly the challenge is we need to know when you do mix and match what parameters you can use and what parameters you can't use, because that's perhaps where the model might fail. But I would say that's our greatest resource is that we can take information from a number of biological systems and how well that does correlate with function and understanding.
Can sometimes, right? So
Yeah.
sometimes you can, sometimes you can't. And I thought you made a nice argument that if the degree to which you standardize is going to make it a lot easier. And I would actually, I think there's another really important thought here about modeling, which is the interface to the experimental community and the idea that models can direct so much experimentation. Yet, are we taking full advantage of that? And I would say no, and I think that's another really vexing problem.
Agreed. Sorry.
Terry Sinofsky, Salk Institute. Just to add another dimension to the discussion about how to disseminate models. So I've noticed that amongst colleagues of mine at the Salk Institute, it's not the PIs who are using the models or the bioinformatic databases. It's the students. It's the postdocs and the graduate students. And that's because they're more computer literate. They have grown up with us, and they've taken courses. And I would like to suggest that what we're talking about here is generational, that we may not be able to get the PIs necessarily to adopt it, or at least they may be encouraging their lab. In fact, some of the faculty are really realizing that without the bioinformatics, they're going to miss the boat. Because they know that their colleagues are having unfair advantages by having access to the databases. But I would say, why stop at the graduate students? Why not push it down to the undergraduate level? After all, I mean, they're even more familiar with using computers and games and things. And it seems to me we ought to be making that available so that young kids could start using the software. In fact, Joel Stiles, who was a collaborator of our lab for many years at the Pittsburgh Supercomputer Center, took his software, MCell and Dream, to high schools, and they loved it. And they're going to be the ones who actually going to be doing the future simulations.
That makes sense, but still, are we realizing as much reuse and understandability of model even to the graduate students and the undergrads as we might? And I would say probably not.
Well, that's a challenge.
Right.
I can't see hands from here, was it? Oh,
Hi, I want to go back to the original question.
Herbert, just say who you are for the record.
Herbert Sauer from Seattle, University of Washington. So I really like Peter's idea of literate modeling. And I work closely with a bunch of pharmaceutical companies on modeling and also academic groups. And the industrial modelers, they do heavily annotate their models. They do almost have literate models, and they have to because they get paid to do that, because they create those models for their clients, and their clients want fully annotated models. Now, we don't have the same reward system, and it goes back to the reward system. We don't have the same reward system in academia for doing that. And we're all on panels, NSF and NIH panels, and we all have to read the data sharing plan. Yes, is there a data sharing plan? Yes, there is. Great. Next. And yet it's never, ever enforced because it can't be. And if there was some extra reward system, maybe you get an extra $20,000 at the end of your grant if you share it in a certain way and you produce a literate model. I mean, if there's no reward system, it's not going to happen.
So maybe I can say a couple of things, share our experience from an undergraduate class here at UCSD, where we actually disseminate what I think are literate models. So I've written some textbooks in LaTeX, and they are actually importable into Mathematica. You can convert all the equations that are in your text into computable cells in that code. And you can actually go through the model step by step, the text and everything. And those must be then, based on the definition you gave, fully literate models, because they're actually out of a book. But they become live in that particular piece of software, and maybe there are other similar pieces of software that can do that. And I'm actually astonished by how much you can do in a software like that. You can take these sliders on the parameter values and just watch the simulations in front of you morph as you change the parameter values. So, and back to Terry's point, I think the undergraduate students just seem to love this because they really sink their teeth into these models, and they can go through them in great detail. But of course, they also learn how difficult programming is and model building is. It takes a long time to do this. So that's our experience here at UCSD.
I mean, we're all in the way you describe what can be done. And we were all involved with journals of some description. There's no reason why journals shouldn't be able to do this. And we are the people who can change that by virtue of the fact we are the customers as well as the people who do the editing and the reviewing and everything else. So, it behooves us to push on that.
And I would say, why not start a modeling section of PLOS Computational Biology where the emphasis is actually not so much on the paper, but on the model, and that your paper doesn't get accepted. Maybe there's a two-page limit on the paper, but the paper doesn't get accepted until the model passes review. And you have to develop what are the review criteria for models, and probably they're going to change over time as we get more sophisticated literate models. But I think we need to move toward making databases and models become published entities and have less emphasis on the papers themselves.
Peter Karp is now the modeling editor of in PLOS Comp Bio.
So this is Chandra Bajaj. I'm from the University of Texas at Austin. And while I liked everything I've heard about reusing models and making it easier to understand them, at least from my perspective, I feel the harder problem that I don't know how to solve yet is modeling error. And that's not to be confused with simulation error. So do we have the right equations that model reality? And how do we go back and validate those models, not verify them through simulations? And quantifying that modeling error is, I think, at the core of anyone who wants to reuse it. I don't want to reuse anything until it's been totally quantified. And so I was trying to throw that to the panel as a question. How much of modeling error is integral into your design principles?
Just want
It's a good question.
Augment that question a little bit, because I've had some private conversations about retraction criteria for models that are either intentionally wrong or are provably wrong, because we retract papers with incorrect or fabricated experimental data with some penalties. And people have raised this question that is similar to the one you're raising. Should we do the same thing in computational biology? That you'll be forced to retract that paper if the model is provably wrong.
Right. Not just the modeling papers.
No, but it's an interesting question. I mean, these standards are very high for experimental work. Should we have the same standards for computational work?
Okay.
So I think the answer to your question really again boils down to the availability of experimental data, because I mean you can check the accuracy of a simulation tool very easily, performing all kinds of test suites but having a computational model with certain predictions or reproductions of experimental data, the question then really becomes, is are the model assumptions correct? Is the model mechanistically correct? But in many cases, the limiting factor is not the simulation technique. I think nowadays we can simulate much more than we have experimental data to support. I think the main bottleneck are the experimental data. And yeah, with higher resolution data, we could more closely inspect computational models, but I think that's the bottleneck. And that's why I also want to bring back this point that we need relatively well-defined biological problems and perhaps even a relatively well-defined biological system, because only by having that, we have a benchmark. How well do our models work together? What kind of interfaces do work, and so on? If we just have a collection of tools, and this tool is applied to this subproblem, but there is no coherence, I think that's dangerous.
I heard you in your presentation. I hear you again, and now I've appointed you as the chair of that session for when we have the open thing. So you'll be able to get all the answers you need, and you'll be able to contribute to it. Other questions?
Microphone back here. Yeah. So, Marianne Martone from UCSD. There's 3 points that I want to pick up on because I think they're very, very important, because this really is about building community. You will see over and over again over the course of these days the dazzling array of things that people have available. And as I was talking to some people at the break, to me the real question here is, why don't we already have a 3D virtual cell, given that we've been able to do these things for a very, very long time? When Christoph Koch took over the Allen Brain Institute and they were doing a connectome, when I heard him talk at a computational seminar, he at Bernstein recognized that the real problem was sociological. It was not technological. At this point, yes, we're imperfect on all of these things, but in fact, it's more of a matter of will and culture. But what Phil said is very, very important, because if you really have a community, the community exerts pressure on each other to do the right thing. And right now we take a very passive attitude, like there's some independent body out there that is forcing all of us to accept all of these modes of publication and other sorts of things, when in fact there's a lot of assumptions about this that says, you know, it's basically us who are creating the students who go to NIH, who go to publishing, who go to the editorship. It's all a circle, right? These are all coming out of the academic laboratories for the most part. And so if we really do want to change, and we all agree that it's good, it needs to actually start to happen. And when you start to look at that sociologically, we hear a lot about the fact that the young are very adept at using computers and share, and that this will all be a sociological change. But academia and academicians are extremely long-lived, and they stay in place for a very long time, and they exert an inordinate amount of pressure from the top. Similarly, though, you know, you need that sort of communication. So I would hope by the end of this, it's not that we've all sat around and talked about the same problems we've been talking about for 20 years, but that, again, if this truly is a community, and we don't know the answer to that, that there will be some recognition that the change is going to have to come from the community. I've seen it with the neuroimaging, where I saw a review of a grant once where somebody said, you did not put your data into this thing. I have been unable to use it. You did not put your tool into this repository which exists. Those have to come from the people who sit on these panels and who review these papers. So, third point, and then I'll give it up, is the point about machine readability. And when we're going to talk about this in the education and dissemination section tomorrow, I really hope that people don't say, oh yes, that's the education and dissemination section that you have to have on an NIH grant, and we're not going to pay attention to you. Because I think the issues of this community building and dissemination really are, can anybody find what we want, you know, what we're doing here, and can anybody reuse it? So when you look at the resource landscape as an ecosystem, you see that you have a fine database and you have a fine tool, but those of us who sit there and catalog these things know there are thousands of fine databases and thousands of fine tools. Putting something in a database does not mean that somebody's going to be able to find it, particularly if the information, as we just heard, is scattered across 50 of these. Why? Search engines can't find what's inside of databases, okay? So this other layer that you have to, first be able to find what's there, and that you have to understand it well enough to be able to reuse it, are three themes that you're going to hear over and over again, and they do require all resource providers and scientists to step back and say, how is my thing going to fit into this ecosystem? Not, listen, you just give me a whole lot of money and I'll solve your problem for you. Because again, we will hear this over and over and over again, and all of us who have been coming to these conferences have been hearing this for a very long time.
Yeah, one thing that might be useful to do is to try to enumerate some of the more specific problems that lack of collaboration is causing. So for example, is it that we need to build huge models and we can't build huge models without having multiple people to contribute to them? That's one possible problem. Another problem is that we need huge amounts of data, and no one lab is going to measure all that data, and so we need a coordinated effort to measure the right data. There's another collaborative problem. What are the other ones?
Tim Clark, Harvard Medical School, and I just want to add something on, again, the literate model thing. I mean, I think it's a common observation that people make, people who develop or use software, that if you hear about a piece of software or a component or what have you that you would like to use, it's developed in someone else's lab or group, and you contact them, what often happens is that if it is reusable, you encounter a lot of issues because they haven't developed, you know, documentation of various kinds. And we were just in a situation like this, and what happened is the documentation gets created on demand, essentially, right? It's what's the word? Well, in the auto industry, but just-in-time documentation, right? So I think obviously there's a question of you know, incentivization, and I like all this stuff about how the sort of the data paper or the model paper that turns things on their head. So instead of writing all your interpretations that you've derived from this data, and the data is kind of, well, here's a couple of images that we made, and then you can get the data set if you ask us, and maybe we'll respond. And so the data is kind of put as an afterthought. Well, let's put the paper as a little bit lower class object. I like all that. But I think the key question in sort of incentivizing the literate modeling or literate data and so forth is that you have to enable the collaboration so that people know that stuff is out there that might be reusable.
Go ahead.
So just to follow back on the
Name and...
Oh, name. I'm Chris McClendon. I'm here at UCSD. Just to follow on the incentivizing, I think incentivizing PIs is very good and a little removed from who's actually doing the stuff. I think undergrads and grad students are very easy to motivate with relatively small amounts of cash or fancy computer hardware, tablets, etc. And I think, you know, something that simple could really encourage data dissemination and documentation and code reusability. Something as simple as just having awards for code documentation within each subfield or something like that.
Yeah, it might be fun to have an award at the ISMB meeting for the most literate bioinformatics program or the most literate model.
That's a good suggestion, James. And then we're going to, and then Terry. Sorry, Terry, then James, and then we're going to motivate you a bit more with lunch.
Great. So Terry Gasterland from UCSD. I'd like to put a bit of a challenge out. As we sequence exomes from hundreds, thousands of people, genomes, one of the things we find is somewhere between 70 and 100 disabled genes due to homozygous or double heterozygous bad mutations. And it would be terrific to be able to take a model off the shelf of, say, a liver cell and plug in the deletion and have the model predict what's going to go wrong. You know, maybe the cell will not divide, and then you'd predict that person doesn't have a liver. But, you know, but more subtly, perhaps cell cycling is slower, or replenishment of something might be slower. And that will then suggest to a physician that there are consequences down the road. And I think then you could also be able to take cells from people who have those issues, grow them up, look at them, and ask, did the model predict properly? There could be something quite simple, but with a huge payoff with something like a liver cell or heart cell model.
So that is a grand challenge. But I would say that now that we put all the protein structures into the E. coli model, we can actually take sequence variations, map it onto the protein, and with computable protein properties from sequence, you can then map them onto the network and then the function of a cell. So in the thesis that was defended here last week, this works for thermal stability of protein, which is one of the more predictable parameters. And if you have enough thermal stability data, you can do that. And then if you, in that case, evolutions in the laboratory to these higher temperatures show up mutations, and then you can map them, and you can actually close that relationship. That is E. coli, liver in a human. I think that is a grand challenge, like I said. But in simpler situations, this is now becoming possible. But only for properties of protein you can actually compute, and as you know, that's pretty tough for most of them.
Last comment before, or question before lunch.
So,
Press the bottom.
But I think your comment that it's a simple thing is illustrative. I think people have to be careful that there's a tacit assumption in the background of many of the things that have been said that we somehow understand all the biology already. And even if you had perfect model sharing, perfect documentation, perfect community, the biology is still hard. And if it weren't, we wouldn't be here. And so to assume that the fundamental problem, when you say, oh, we're going to take these things to undergraduates and they're suddenly going to be able to play with sliders and understand things, I think that that's a very dangerous thing if you think that the technology is going to somehow provide understanding by itself. Fundamentally, the best thing we can do is to make sure that the problem is where it should be, which is understanding the biology. If we do our jobs right, then people can concentrate on the hard problem, which is understanding the biology. But no matter what we do, we are not going to make the understanding of the biology less difficult, because the biology itself is difficult to understand. And so that I think is a key point. The other thing is that I think we have to be a little cautious, precisely for that reason, when we talk about, in a very generic way, about the data, the models, the experiments, as if we were talking about some kind of well agreed upon homogeneous set of describable processes. In each subdomain, in each particular problem, those terms are going to mean very different things, and you may not have a shared description that will encompass all of those. And so I think you have to be very careful both about thinking that the biology is simpler than it is, and about thinking that the heterogeneity of biology can be necessarily reduced easily. And you may need many levels of infrastructure and many levels of abstraction and many details. As my friend Marcelo Magnasco at Rockefeller likes to say, nature has a criminal mind. For every biological phenomenon and rule, you will find a counterexample. And so we have to be responsive to that variation and that richness and that difficulty, which is intrinsic to the biology, and no amount of computational infrastructure can reduce that complexity.
Here, here. I think what we're going to formulate a few out of this, and I think it's going to be actually pretty straightforward. There won't be enough, but we can certainly formulate a set of topics to be discussed in the open session later on. But if you have strong feelings about that you and a collective, a group of people would like to discuss, then do let us know. We will have facilitation to help with this process, and you will be able to go from you don't have to stay in one place for the whole time. You can go around to other places. But I think there are lots of really interesting questions and challenges that have come out of this session this morning. And I think it's hopefully you'll all participate in discussing them more, and hopefully, and I take Marianne's frustration at the end, and I think some real action out of this is what we're looking for. So, but before that, let's have lunch. Okay. Enjoy. Thank you for the speakers.